Today we will discuss. It is visible to all of you.
Yes, sir.
So this is all about your anti-diabetic agents. So for this you have to discuss about your endocrine system or endocrine glands like pancreas. Okay.
Yes, sir.
Wait just for a minute. All about your anti-diabetic agent. So for this you have to okay. So this is all about your pancreatic systems. It is one of the gland which secrete your pancreas. Okay. Sorry, pancreas is the gland which secrete your insulin hormone. One of the endocrine. Pancreas is one of the endocrine system which can secrete the hormone like insulins. So insulins are mainly responsible to maintain the blood glucose level. Okay. So screen is visible now. So that glucose, if the glucose level is increased in the blood, that is called hyperglycemia. Hyper means increase in the level of glucose in blood. Glycemia means glucose in blood. Hyper means more. Or level of glucose is more in the blood is called hyperglycemia. So if the pancreas is unable to secrete the insulin properly, due to some disorders, maybe autoimmune disorders or any pancreatic disorders, then insulin secretion is very less. So that automatically glucose level will be increased and it will be causing some type of disease called diabetes. You are telling diabetes means increase in blood glucose level. That term is called diabetes or hyperglycemia. So that you have to control the level of glucose in blood by using some anti-diabetic drugs, or you can go for some healthy diets. Okay, not like your other junk foods. You have to take some good or healthy foods, or you have to go for exercise. So diet control and exercise along with medications is more preferable. So you have to maintain the blood glucose level by different methods, maybe anti-diabetic drugs, or sometimes you can use some insulin preparations. So normally or naturally it is produced in the pancreas. Again you have to give, it is not controlled suppose, you have to give from outside through injections. Insulin preparations are available nowadays. So they may be extracted from the human or maybe dog or some other sources. Commercial sources are available. So one by one we'll discuss how the insulin is regulating the blood glucose level. Okay. So diabetes is a disease state which impairs the body ability to maintain blood glucose or blood sugar level. So in the stage when the body is unable to maintain the blood glucose level, that is called diabetes. So it is a disease state when the body system, your body is unable to maintain the blood glucose level or blood sugar level. Okay. So it is otherwise called diabetes mellitus. It is a group of metabolic disorder Why it is called metabolic disorder? Because your insulin is directly responsible for metabolism of glucose. So if it is not able to metabolize, then you can say that it is a metabolic disorder. So it is a one form of metabolic disorders. It is characterized by hyperglycemia. Hyper means the level of glucose in blood is more, and of course due to defects in the secretion and action of insulin. So the secretion of insulin may be decreased, or the action of insulin may be also decreased. So that will cause directly the increase in glucose level in blood. Okay. So the secretion is decreased by any infection of the pancreas or any autoimmune disease. Okay, and the action may be sometimes decreased, so that the insulin is unable to control the glucose level. So by these two methods, your level of glucose is increased. So based on that, you can say the diabetes may be different types. One is type 1 diabetes, another is type 2 diabetes, another is called gestational diabetes mellitus. It is mainly in pregnancy time. So there may be imbalance of glucose level in blood during pregnancy. So there may be some physiological changes there. That's why the glucose level is unable to control. So that is called gestational diabetes mellitus. This is some other types. So generally you have type 1 and type 2. In case of type 1, it is depend on the insulin. That is called insulin dependent diabetes mellitus. Type 2 is non-insulin. Means it is not depend on the insulin, but the insulin is unable to control. Here, this is secretion of insulin is not present. It is very decrease in the secretion of insulin level. So that is unable to control the glucose level. So one by one we will discuss. Next page. You see, type 1 diabetes mellitus or diabetes can develop at any age, but mainly it occurs in children and adolescents. So up to 18 to 20 years age, or during children's, it is more frequent in case of type 2, type 1. So it is in case of type 1 diabetes. So mainly pancreatic beta cells are there. So the cells present in pancreas. So in pancreas, the isolates possess beta cells, and that beta cells are able to secrete your insulins. Okay, that beta cells are mainly present inside the isolates of pancreas and responsible for secretion of insulin. If these are damaged or destroyed through autoimmune systems or autoimmune disorders, so that pancreas is unable to produce sufficient insulin to control the blood glucose level. So here you see this is the normal beta cell or normal isolate. Inside beta cells are present. You see the colors, beta cells. So after damage of these beta cells, it will be converted to like this. So color is changed here. These beta cells are destroyed. So if these cells are destroyed, they are unable to produce sufficient quantity of insulin. So that automatically it is not able to control the blood glucose level. As it depends on the insulin, that's why it is called insulin dependent diabetes, because there is no sufficient insulin. So they are depend on the insulin. So it is called insulin dependent diabetes mellitus or juvenile onset diabetes. Juvenile means in the your children's stage or child stage it is showing. That's why it's called juvenile onset diabetes. You can see here diagrammatic. So here you have normal beta cell. You see the color here. Blue colors are there. Here it is changed to pink color. So these beta cells are damaged. So by this way, so how beta cells are damaged? By different disorders like autoimmune disorders. So that it is unable to control your blood glucose level. Okay. So this is about your type 1 diabetes. It is called insulin dependent diabetes. It depends on insulin. Okay. Next we'll go for type 2 diabetes. It mainly occurs in the adults. Okay. So after 30 years age, you can get the diabetes like type 2 diabetes. Okay. It is characterized by insulin resistance and called as non-insulin dependent diabetes mellitus. Diabetes mainly occur in adult stage. That's why it is called adult onset. Why it is called non-insulin? Because there is insulin resistance. So the insulin is unable to control the metabolism, or they are unable to control the metabolic process of your glucose. Glucose is unable to absorb the insulin. I'll show you the diagram how resistance is developed. So due to that, it is not depending on the insulin. That's why it is called non-insulin dependent. But in first case, it depends on the insulin. Okay. But in second stage, in type 2, the insulin is totally unable to absorb by the glucose, so that glucose metabolism is not there. Okay. So people with type 2 diabetes, you have family history of the condition. If anybody in your family, your parents or grandparents have type 2 diabetes, so you may get some diabetes in your adult stage. Okay. So that you have to maintain your diet and exercise properly after 30 years age. Okay. Another one is gestational diabetes. It involves high blood glucose level during pregnancy, and the complication is associated with both mother and child. So in both cases, so it will cause some increase in glucose level due to the change in the physiological system during pregnancy. Okay. You see the diagrammatic presentation how type 1 and type 2 involved or diabetes is caused due to different mechanisms. So in case of type 1, only the beta cell is destroyed and insulin secretion is very less, so that there is increasing glucose level in the blood. Okay, that is type 1. But this type 2 is mainly involved due to different metabolic process. This metabolic process are involved in different parts of the body, or and in this area or in the part of different parts of the body, there will be more glucose level, so that it is resistant. So the insulin is resistant, so that it is unable to absorb by the glucose for its metabolic process. Okay, so this is the difference in type 1 and type 2. You see, in case of type 2, you see how the resistance is developed. So in normal stage, there is pancreas and it secretes insulin. More number of insulin is secreted. And whatever the glucose is there, automatically you see insulin is moves glucose to the cell. This is your cell. Okay. So along with glucose and insulin, it is entered or absorbed by the cell. So that metabolism takes place. But in case of diabetes, what will happen? Pancreas produce less number of insulin or less volume of insulin, and but glucose level is increased here. So what will happen? It is unable to metabolize or unable to cross the cell or absorbed by the cell. Here you see, insulin moves less glucose to the cell. So both glucose and this one less absorbed, so that there is less metabolism of glucose. So it is totally resistant. Means due to less number of insulin, it is unable to metabolize. Again, it is not absorbed properly by the tissue or cells. So that metabolism of glucose is very less. So this is about type 2. Okay. So insulin resistance is developed in case of type 2. You remember. Means this insulin is not able to move inside the cell properly for the metabolism of glucose. Okay. And there is more level of glucose here in case of diabetes. Okay. You see the causes and symptoms. What are the different causes of diabetes? So you may have genetic defects, or genetically it may be inherited from your parents, or there may be some pancreatic disease, okay, or you may have overweight. So as per your age, your body weight should be maintained by your exercise and diet. So it is most required. Or you may have some stress, so that will also increase your, or you may have some damage of the pancreatic system. Okay, and some other medications or due to their side effects, there may be problem in the pancreatic system. So that insulin secretion will be very less. Okay, and what are the symptoms? So you may have some urination, frequent urination will be there. So you will go for urination number of times. Tingling in the hands. So in hands you will get some spots. And extreme fatigue, you will not feel, means always you will feel tired. That is fatigue. Increased thirst, you will feel thirsty. Okay, next you can feel hungry also. Slow healing of the wounds. Any wounds in the any part of the body, it is slowly healed. Okay, and blood vision. I blood vision will be there. Okay, you are unable to see properly. There may be weight loss. So many things are there you have to maintain properly. If there is any signs and symptoms, so you have to check your blood glucose level and go for treatment. So what are the treatments? First you try with your controlled diet, that is calculated diet with exercise. Don't take any medications first time. So diet control with exercise is must. If it is not controlled, you have to go for any test again, blood glucose test. If it is not, it's showing any glucose level is more, then you can go for oral medications or anti-diabetic drugs. If it is not controlled, suppose, then you can go for insulin preparations. Okay, so by this way you can confirm. So you can go for some home glucose monitoring now, testing possible in your home also. You can go for testing. And you can go for regular exercise. So these are some of the guidelines under diabetes. So what are the diagnostic tests you will perform and what are the readings you will get? From that you can confirm whether you have diabetes or not. Okay. So you can go for fasting group test, FPS, fasting blood sugar test. So in that case, before taking any breakfast, you can go for test. So you should have normal glucose below 100. Okay. If it is the, if this value is showing, you can say it is a normal. If it is 100 to 125, it is showing that it is a pre-diabetic stage. It means it can go for diabetes next stage. If it is more than 126, it is diabetes. So it will fully confirm that you have diabetes. So by this way, or you can measure in terms of millimole per liter, 5.6, and these are the 5.6 to 6. Like this also you can measure, or you can go for mg per deciliter. So any form you can measure your blood glucose level. Again you can go for postprandial, means after taking food, what is the level of glucose in blood? That is after 2 hours you can check. So if after 2 hours it is less than 140, then it is normal. If it is again more than 140 to 199, you can say it is a pre-diabetic state. If it is more than 200, it is diabetes, fully confirmed. Okay. So these are normally you can on the initial stage. Suppose you have severity in the diabetes, you can go for like this HbA1c. So they will check in the molecular level how the metabolism takes place. So these things, if it is less than 5.7, then it is normal. 5.7 to 6.4, it is pre-diabetic. More than 6.5 is diabetic. So you should confirm from these readings. Okay. So you see, you can see the diagrammatic presentation. So you should be fasting for 8 to 12 hours before going for any test. Fasting blood sugar means at night time you should take at 10 o'clock. Next day morning you have to take the, you can go for the test. So that is called fasting blood sugar test. So if it is less than 100, it is normal, green sign. Then you will go for yellow region. If it is pre-diabetic, if it is red area, it is diabetic, fully diabetic, more than 126. So you have to maintain the blood glucose level by your diets and exercise. Or finally, if you are interested for molecular level test, you can go for HbA1c test. Okay. Same thing I have discussed. What is the normal stage, pre-diabetic stage, and diabetic stage. So like this. So after your food, that test is also called as oral glucose tolerance test. This after taking food, how much glucose level is there. That is called oral glucose tolerance test. Okay. So these are all about your diagnosis. Okay. So for treatment, what you can take, what is the requirement. So these things we will discuss one by one. So first case, you have to go for diet and your control exercise, then anti-diabetic drugs. If not possible, then you can go for insulin preparation. So as per syllabus, it is given insulin preparation. That's why I have given here first. But first you have to start with diet exercise, followed by your anti-diabetic drugs. If not successful, then you can go for insulin preparations. So insulin was first discovered in 1921 by the scientists Venting and Best, who demonstrated the hypoglycemic activity of pancreas extract from the dog. So from the dog, they have extracted the insulin. From the dog pancreas, they have extracted the insulin. So first they tried in 1929. So what is the insulin structure? As a chemistry student or pharmacy profession, you should know what are the structures, how they are interlinked, how they are involved in the glucose metabolism. All these things you have to know. For GPAT exam also it is required. So it is a polypeptide, means it is generally a protein in nature. A number of proteins are linked to produce polypeptides, or it is a polymer of amino acids. Okay. So how many amino acids are there? There are 51 amino acids with molecular weight 6,000. Okay. So there are two chains. One is chain A and another is chain B. So chain A is having 21 amino acids, chain B is having 30 amino acids. So both chains are linked by disulfide linkage. Double S is there. So I'll show you the diagram how they are linked. Okay. Disulfide linkage. Okay, there are minor difference between human, pork, and beef insulin. So from different source you are getting the insulin. So maybe from directly human or pork or beef. So pork insulin is more homologous to human insulin. Means both insulins are having similar sequence of amino acids as compared to beef insulin. So that's why pork insulin is more extracted commercially. Okay. So insulin is synthesized in the beta cell of pancreatic islet. So it is naturally produced in the human beings or other animals also, like pork or beef. Okay. So I'll show you the structure. You can see. So this is A chain and this is B chain. So both A chain and B chain are linked by disulfide linkage. Okay. So this is also a diagrammatic. So numbering will start from here. So in case of amino acid, there are two groups, amino group and carboxylic group here. So this is amino group one end, this is glycine. So glycine is one end, okay. And this side, chain A, asparagine, okay. So you can number from here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20 and 21. So A chain is total 21. Next B chain is 30. You can count from here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30. Okay. So THR means 309, lysine, proline, 309 again, tyrosine, phenylalanine, glycine, arginine. So all amino acids are involved here. So if we go for 3D structure of this insulin, so you can see here C represents this carboxylic acid part, C and C. Carboxylic acid part. N and N means NH2, NH2. This part. So this is the 3D structure and this one is your 2D structure. Okay. So there are total amino acid present in the, your insulin is 51. Chain A is 21. Chain B is 30. So you remember for your exam. Okay. So this is your structure of insulin. So what are the characteristics of the insulin? So based on this characteristic, you can categorize the insulin. So first is onset, means how quickly they act. That is the onset of action. Okay, maybe 1 hour, 2 hours, or 10 minutes or 15 minutes. On how quickly they are producing their actions. That is called onset. Based on onset of action, there are different types. Okay, peak means how long it takes to achieve the maximum impact, means to produce their action. What is the level of insulin in blood? That is called peak. Okay, level of insulin in the blood is called peak. Duration, how long they last before they wear off, means how much time they are producing their action. It's called duration. That is called duration of action. And concentration of insulin, how much concentration of insulin is active. At what concentration they are producing their actions. That is called concentration. And route of delivery or administration. So they may be injected under skin or they may be given intravenously. So based on route of administration also, there are different types. So these are different characteristics of the insulin. Okay. So human insulins, you can use directly human insulins or you can use insulin analogs. They are available from the other sources. Are available for insulin replacement therapy. If your pancreas is unable to produce insulin, then you can use this type of insulin. That is called insulin replacement therapy. You can replace the original insulin secreted naturally. Okay. Insulins are classified by the timing of their action, maybe short acting, long acting, intermediate acting, or fast acting, depending on the duration or time of their action. There are different types. So here there are different types. One is fast, intermediate, long acting, and sometimes short acting is also there. Depending on the duration, there are different types. So first one is fast acting insulin. So fast acting means these are absorbed quickly from your fat tissue. Means you are administrating in the subcutaneous layer. Under the skin you are administrating the insulin. So from your fat tissue they are quickly absorbed into the bloodstream. That's why it is called fast. It is used to control the blood sugar during meal and snacks and to correct the blood sugar, high blood sugar. So if you are taking, so these are mainly taking during meals and snacks. Suppose if you take meals and snacks, during your glucose level is more. So that time you can use this type of insulin to maintain the blood glucose level. Okay. Next is fast acting insulin may be different types. One is rapid acting insulin. Rapid acting insulin means it will give its action very rapidly. So you have different types under this: insulin aspart, insulin lispro, insulin glucoside. So you remember these terms. So these are different types of rapid acting insulin analogs, which have onset of action of 5 to 15 minutes. So within 5 to 15 minutes they will produce the action. So very rapidly they are producing the action. And peak effect in 1 to 2 hours, means they will reach the highest level in blood in 1 to 2 hours. So first they will effect start from 5 to 15 minutes and they will reach up to 1 to 2 hours. Okay. And total duration of action lasts for 4 to 6 hours. Up to 4 to 6 hours they will give their actions. They will continue up to 4 to 6 hours. This is called a rapid acting insulin, and these are the different types of rapid acting insulins. You remember insulin aspart, insulin lispro, insulin glulisine. So you will get in your exam short notes insulin and its preparation. So you have to write different types with examples. Then another is regular human insulin. It is having onset of action half an hour to 1 hour. So within half an hour to 1 hour, it will give the action. Peak effect is 2 to 4 hours. So up to 2 to 4 hours, it will reach the highest level in blood. And duration of action is total duration. It will produce 6 to 8 hours. So the larger the dose of regular, the faster the onset of action. If you increase the dose, it will give more onset of action. And but the longer the time to peak effect, longer the duration. So if it is taking more time to reach the peak level, then it will give the duration of action for a long time. That is the meaning. Okay. So this is the things. Intermediate acting insulin. So what is intermediate? Is they are absorbed and last longer. Means they are producing the action for longer time. Okay, but absorb slowly. So if it is absorbed slowly, it will give slowly the activity for longer time. That is the meaning. Okay, it is used to control the blood sugar overnight. So total whole night it will maintain your blood glucose level while fasting and between meals. So fasting means in night time we are not taking any food. Suppose night 10 p.m. to morning 10 p.m. So night 10 p.m. to morning 8 a.m. So that is fasting period suppose. So at that time it will maintain and between meals. So morning, if night 10 to morning 10, suppose you are not taking any meals. So during that period it will control the sugar level. So based on the severity and condition of patients, you have to take your different type of insulin. Okay. Then is intermediate acting insulins. Intermediate. Okay. So this is intermediate these terms, and it is again two types. One is NPH human insulin, NPH, which has onset of insulin effect on 1 to 2 hours. So within 1 to 2 hours, it will give the effect. And peak effect is 4 to 6 hours, and total duration is more than 12 hours. Means total time is increased here, the total duration. Very small dose will have earlier peak effect and shorter duration of action. Okay, if you are giving small dose, it will give earlier peak effect and more shorter duration of action. If you increase the dose, higher the dose, it will give longer time to peak effect and prolonged duration. Duration of action is prolonged or increased. Another is pre-mixed insulin. What is pre-mixed insulin? Which is NPH pre-mixed with either regular human insulin or rapid-acting insulin. So they are mixed type. Means you can mix the NPH type of insulin with regular insulin or rapid-acting insulin. So anyone you can mix. This insulin action profile is a combination of short-acting and intermediate-acting. So both are mixed here, short-acting and intermediate-acting. Okay. Next is long acting. It is absorbed slowly, minimum peak effect, and stable plateau effect that lasts mostly day. So it will give the action for a whole day. That's why it is a long acting. Okay. So it will give a plateau stage. Okay. It is used to control the blood sugar level overnight. Okay. So overnight you can maintain the blood glucose level during fasting and between meals. So it will maintain all the stages overnight, during fasting, and also between meals. Means night 10 p.m. to morning 10 a.m. So total things it will maintain. So that's why it is a long acting. Okay. Then long acting insulins may be different categorized. First one is long acting insulin analog. It is examples given: insulin glargine and insulin detimir. So these are two types of long-acting insulin which have onset of effect within half to 2 hours, half an hour to 2 hours. The insulin effect plateau over next few hours. So after pause it will be constant and followed by relatively flat duration of action that lasts for 24 hours. So it will continue for a whole day. Okay. So it is a long acting. You can see the tabular form of this onset, peak, duration, and appearance. So different types. One is fast acting. You have Lipspro, Aspart, Glulicide. So how they are giving their onset of action, peak, and total duration. Intermediate, it is having NPS type. Long acting, you have Detimir and Glaragine or Glargine. So based on that, you can remember for your exam in tabulated form. Then what are the main type of insulin preparations available in your market? Same thing I have discussed. So another tabulated form it is given. So some are given lenti, semi-lenti, ultra-lenti. I will discuss on next slide. So these are the marketed formulations available for the treatment of diabetes. Okay. So what are the preparation of insulins? So the older commercial preparations are mainly produced from beef and pork pancreas. So commercially they are giving more volume of insulins from this pancreas. That's why they are using commercial. They contain 10,000 ppm of other proteins, proinsulin, other polypeptides, pancreatic insulin. Is there commercially available in more quantity, okay? And they are potentially antigenic in property. They are no longer produced and have been totally replaced by high purified pork, beef insulin and recombinant human insulin. So nowadays these techniques are not used. Nowadays we have recombinant DNA technology. So that we are applying in human beings also to get some more quantity of insulins. That is recombinant DNA technology. And you can see this is another preparation. It's called highly purified insulin preparation. So in 1970, we have used some purification technique like gel filtration, ion exchange chromatography. Okay. So these are mainly applied to produce monocomponent insulins, which contain less than 10 ppm proinsulin. It is a very purified. Previously it was 10,000 ppm. Means they are mixed with some other proteins like proinsulin, polypeptides. So it is difficult to separate. So purification is difficult. Now it is very purified insulin. So you will get very original type of insulin. So it is mainly purified by gel filtration and ion exchange chromatography. So in your analysis, you can read your chromatographic methods. Gel filtration, ion exchange chromatography. How can you purify the biological samples like insulin or other type of hormones? So they are less antigenic. Means they are not harmful for the body systems. Less antigenic. Okay. You can see the some drugs available in the market. Rapid acting insulin. Under that you have already naming is given insulin aspart, and the brand name is Novolog. Novolog. It is same with regular human insulin except that proline. So proline is one of the amino acid present in the chain system. It was replaced by aspartic acid in position B28. B28 means in beta chain 28 position, this proline is replaced by aspartic acid. So that you will produce the insulin aspart. It is a synthetic type, means it is derived from the other sources and similar to human insulin, only the change in proline. Proline is replaced by aspartic acid in position 20. You remember, in case of insulin aspart, which is replaced by aspartic acid. So proline at what position? B20. And your onset of action is very quickly, 15 minutes. And duration is 4 hours. Okay. So this is related to insulin aspart. So you see, remember what are the amino acids changed. Here, another is insulin lispro. The name is Humalog. So here you see beta chain 28 position lysine is there, beta chain 29 position proline is there. So these are human insulin analog where this proline and lysine is reversed. So 28 position lysine is there, it will be proline. 29 proline is there, it will be converted lysine. So the position is changed. Okay. So due to structural changes, there will be change in the activity. It may be very potent sometimes. Okay. So the total normal human insulin structure is changed to produce the synthetic products. Okay. So here onset of action is 15 minutes, duration is 2 to 4 hours. So you remember insulin lispro, what are the amino acid replaced? Insulin aspart, what is the insulin changes? Next we'll go for insulin glucoside. So in this case, asparagine at position B3 replaced by lysine, and lysine at B29 is replaced by glutamic acid. So there is change in the amino acid sequence. So it is same with other rapid acting insulins. So you remember all amino acid sequence, how they are changed and getting the activity. Regular insulin, it is called Humulin. It is available in the market, otherwise called Novolin. The only insulin that is approved to be administered by IV route. Remaining are subcutaneous, this is intravenous route. It comes from pork, that is called porcine, from beef, it is called bovine, or human pancreas, or genetically engineered using recombinant DNA technology from certain microorganisms like E. coli. Okay? So from different sources, these regular insulins are extracted, and this is only approved for IV route. Answer is 30 to 60 minutes, duration is 6 to 8 hours, total duration. Then we will go for intermediate type of insulin. So your name is given isofen insulin, or it is otherwise called neutral protamine agonist, or NPH, or otherwise called humulin N and novolin N. Okay, N represents this one, NPH. So this is a neutral protamine agonist. So it is the sterile suspension of zinc insulin crystals and protamine sulfates. Okay. So it is a protein from the sperm of fish. That fish name is given here. So it is a combination of zinc insulin and protamine sulfate. Okay. So if it is a coming question, so NPH is a combination or sterile suspension of zinc insulin crystal with protamine sulfate. And this protamine sulfate is the protein of sperm of fish. Okay. The onset of action is 1 to 2 hours. Total duration is 10 to 16 hours. So this is the intermediate type of insulin preparations. Another is insulin zinc suspension, that is called lentic, otherwise called lentic. Insulin zinc preparation is a sterile suspension of insulin modified by the addition of zinc chloride. So it is a insulin preparation and it is modified or attached or bind with zinc chlorides. Okay. It has the advantage of fewer allergic reaction because it is given from external source. So that's why it may be sometimes antigenic type. So there is less allergic reaction with NPH. Onset is 1 to 2 hours. Total duration is 10 to 16 hours. Then long acting. Long acting you will get glargine. Glargine otherwise called Lantus. It is differ from the human insulin that asparagine at position A21 is replaced by glycine and additional some other amino acids are also attached. Okay. So it is a clear solution and it will give the action up to 24 hours. That's why it is a long acting. Okay. Onset is 2 hours, total duration is 24 hours. So this is the duration of actions. Another is extended insulin gene suspension, that is ultra lengthy. Ultra lengthy means it is extended for some more time. Means duration is 36 hours. Previous is 12 to 24 hours. It is 36 hours. So duration of action is extended. That's why it is called extended insulin gene suspension, ultra lengthy. It is a sterile suspension of insulin modified by the addition of zinc chloride. So zinc chloride is added along with insulin. This is called long acting or ultra lengthy, means extended. The action of duration of action is extended. Another last one is long acting type insulin detimir. It is another long acting insulin that may have some benefit over isofen insulin. It is a neutral insulin, human insulin, in which the terminal amino acid at B30 is replaced by myristic acid. Another fatty acid is changed here. Instead of amino acid, they have used protein fatty acid, that is myristic acid, so that they got some new activity, or they may got some long duration activity of insulins. So up to 24 hours they are giving the activity, that is insulin detimir. So here only this terminal amino acid at P30 is changed by this myristic acid. Okay, it is another important question you may get. This is only fatty acid change, not amino acid. Okay. Then sometimes insulin combinations are also available in the market. That is pre-mixed insulin. Okay, one is novolin 70-30. These are the different doses. Content 70% NPH with 30% regular insulin. This is a combination. Okay, another is humulin 50-50. Contains 50% NPH and 50% regular insulin. Okay. So these are the combination of insulin. So depending on the severity of your patients, you can use this type of combination therapy. Mixture of insulins are being prepared prior to administration if the pre-mixed combination of insulin are not suitable for the patient or not available. Okay. Sometimes if not suitable, then you can use some other combinations. The combination of either rapid or short-acting insulin and intermediate or long-acting insulin being used. So it is given the regulation. So either you can use combination of rapid or short-acting or intermediate or long-acting. Except insulin glargine, which should not be used in combination with any other type of insulin. So they are giving some allergic reactions to those, it is not useful. So you have to use any option, rapid or short acting, intermediate or long acting. You have to use combination. So these are the things given under your insulin and its preparation. So you can go thoroughly. Another is guidelines in mixing the insulin prior to administration. So before administration of insulin to the patients, you have to go for some guidelines. If not specified in doctor's order, use insulin of same concentration. So whatever you are using regularly, you have to use. Otherwise, you don't change the concentration. Use proper syringe to measure insulin in units or ml. How much ml you are giving, you should use the syringe, insulin syringe. Don't contaminate the content of one vial with the content of another vial. So don't mix any vials. Okay. So these are the guidelines. Always draw the short-acting or regular insulin which is clear, fast, then follow it the intermediate or long-acting. Okay, so mainly the regular insulin is clear. There is no cloudy, clear solution. So first you give this one, followed by your intermediate or long-acting, because these are cloudy. If you use first this one, then syringe will be used for this regular. So this regular clear solution will be again cloudy. Okay, so first you give regular insulin or short acting insulin, followed by your intermediate or long acting insulin. If you are using any combination, okay. Never mix a short acting insulin with lengthy or ultra lengthy. So this is the guideline. Never mix phosphate buffer insulin, that is isofen insulin, otherwise called isofen, because phosphate is buffer with the insulin, isofen, with lengthy or semi ultra lengthy. So they never mix, don't mix the isofen insulin with lengthy or ultra lengthy. So these are the guidelines you remember for your GPAT also. And these are some of the same things I have given in a summary. So you can read for your exams. What are regular insulin, lengthy insulin, isofen insulin, and what are the different preparations available in market. All these things I have given here. So I'll send you the material, you can read thoroughly. So this is your total insulin preparations, okay. Means these are some extra materials you can read for your GPAT exams. Where the different humulins are available. Humulin R, humulin L, L represents lengthy, humulin N, okay. So there are different types of humulins available in the market. Then what are the reactions? Suppose you are giving the injection at a particular site of your subcutaneous part. So there may be some lipodystrophy. Lipodystrophy means, so there will be you are giving the injection at same site. So, so this condition is called lipodystrophy. So this is the reactions or allergic reaction for this site. So if you are giving the injection continuously on this fat area, so it may have some reaction. So it is called lipodystrophy. And some other things like insulin facilitates glucose transport across the cell membrane or skeletal muscles, fats. So these are the other actions of insulin. So it will mainly facilitate the glucose metabolism through different parts like skeletal muscles, cell membranes, and fats. Insulin facilitates glycogen synthesis from glucose in liver. So if it is glycogenesis or glycogenogenesis, yes, synthesis of glycogen from the glucose. So that glucose level is automatically decreased. So this is the role or mechanism of insulin. So it may be in liver, muscle, fat by stimulating the enzyme called glycogen synthase. So in presence of glycogen synthase, okay, there will be synthesis of glycogen from glucose. So that automatically glucose level is decreased in liver, muscle, or fat. Okay. Insulin inhibits gluconeogenesis from protein, fatty acids, and glycerol in liver. Okay, inhibits means gluconeogenesis means it will cause formation of glucose. So it is inhibited by the insulin. We want only the decrease in glucose level. So it is inhibiting glucose synthesis. That is gluconeogenesis. Glucose synthesis is inhibited. Okay. These are the action of insulin. Insulin inhibits lipolysis. Lipolysis means again lipid is converted to glucose. So it is inhibited by insulin in adipose tissue and favorites the triglyceride synthesis. Triglyceride is required for your body system. So these are the different process by which your insulin is required for different metabolic process. Insulin facilitates amino acid entry and their synthesis into proteins as well as inhibit protein breakdown in muscles. Okay, insulin deficiency leads to protein breakdown and automatically it is increasing the glucose level. So your aim is to decrease. Okay, so it should inhibit the protein breakdown in muscles, so that there will be less glucose level. Okay. Fate of insulin. How insulin is metabolized is distributed extracellularly because you are giving in the subcutaneous part. So it is a peptide gets degraded in GIT if given orally. If injected insulin, so it is metabolized in liver and to a smaller extent in kidney and muscles. So general metabolism takes place in liver, but if it is given orally, then it is degraded in GI system. Okay. Then next class will go for anti-diabetic drugs. So these are all about your proteins, insulins and different insulin preparations, mechanism of insulin, metabolism of insulins. So all these things you go one by one. I'll send you the material. So if you have any doubt, you can ask me any question now, and I'll send you the material. Go thoroughly, all materials. Okay. So these things I'll discuss on next class. Any doubts you can ask me now. Give your presence one by one, or anybody have any doubt? So any two topics are left on the 4. I'll send you the material. Same thing already I have discussed. So I'll send you the material. You read. There is no such important things are there. Already I have discussed. I'll send you the material. You go the material and read. Any doubts you can ask me. Okay. Tomorrow in my class, Deepak sir will take your industrial pharmacy, 3:30. So you should be present that time. So nobody has any doubts? So we'll conclude today this class. So you read thoroughly. I have already sent you material previous class. You read it. Any doubts you can ask.